talking about, I'll be talking about MARTINI coarse-graining for molecular dynamic simulations. So I'll be only talking about how to coarse-grain a protein, but this can be extended to lipids and other biomolecules, even the DNA. Coarse-graining is what I'll explain in the next slide. Why do we coarse-grain is because instead of atom-to-atom calculations, in the coarse-graining approach, we combine 3 to 4 atoms into a bead. So that reduces the calculations by 2 to 3 orders of magnitude. So generally, atomistic simulations take longer time, or they're expensive in terms of computational efficiency, but coarse-graining reduces that, but it's always a trade-off between accuracy and efficiency. So it also serves to bridge the gap between the atomistic and mesoscopic scales. For example, if you consider cellular processes, they cover time scales and from nano to microseconds to seconds to milliseconds, and the length scales are like micrometers to meters. So let me just explain how the coarse-graining works. Coarse-graining approach here is to map the atoms. Generally, we consider 4 atoms. For example, if we consider this part here, we combine OH, carbon, carbon, OH as one bead, 4 atoms. And this 4 atoms is one bead, as shown here. But ring structures is tricky. Instead of combining all the 4 atoms, we just take sometimes 2 atoms for maintaining that structure. So we have only 2 atoms represented here as TC4, which is like a smaller bead. And generally 4 atoms or 2 to 3 atoms as one bead. Yeah.
So yeah, and coarse-grain rings up. So you found those beads there. So do you put any physical constraint that?
Yeah.
About the shape of the ring?
Yeah, we do. In case of protein, we use secondary structure depending on where the molecule, where the amino acid is present. We impose the bond lengths and dihedrals based on where the amino acid is present in the secondary structure. If it's in helix, it's a different model compared to if it's in loop or bend, it's a different thing.
So are the positions of these nodes, if I may call them, are they determined with the center of mass?
Yeah. Center of mass. So the 4 atoms in the center of mass, including hydrogen atoms in the MARTINI 3, but this paper is for MARTINI, I think 2.1. Yeah. So another interesting thing about coarse-graining is it also retains some chemical specificity. For example, the model considers mainly polar, apolar, charged, and non-polar interaction sites. So also within each subtype of these, they can be distinguished. We can distinguish between hydrogen bonding capabilities. For example, donor acceptor have different chemical groups. And the degree of polarity is also varied from P1 to P5, with P1 being lower degree of polarity compared to P5.
One thing is also, okay, go ahead. Yeah.
Oh yeah, So this is like an interaction matrix, right? Like where how we assign the charged or polar or intermediate is non-polar is generally what they consider, and apolar groups. So depending on, so DA here indicates hydrogen accepting capability, and D is donor. I think I might be wrong, but D is the donor, A is the acceptor. TA is both.
Yes.
Sorry, TA is both. So depending on that, we assign interaction matrix, which we use to calculate how each bead interacts with the other. So how the parameterization is done? In this case, the strength of interaction is determined by, for example, if you consider non-bonded interactions, so that is generally derived from Leonard-Jones equation. So how the strength of interaction is determined by this value of depth here. So for hydrophilic, if the amino acid is hydrophilic, it has higher EIJ, this depth here, and it has lower for hydrophobic amino acid. So, so those are the values, for example, 5.6 kilojoules per mole for hydrophilic amino acids. So what, by essentially considering different depths, we can mimic hydrophobic effects. For example, some amino acids which are hydrophobic in the solvent environment like water would want to stay inside, right? So to mimic that effect, we consider the depth. So how LJ interactions are parameterized is by reproducing the experimental partition-free energies. For example, if we mix the amino acids in water or in butanol or octanol, we calculate the equilibrium densities of these beads in oil phase and in water phase and calculate delta G. So we try to match delta G from the calculations, like long enough MD simulations with the experiments, and parameterize or iterate them until they match with the experimental, in terms like reduce the error between the experimental delta G versus the MD values. So moving on, there's another term in non-bonded interactions, that's the Coulombic term. I think in general force field, we remember there's bonded interactions, non-bonded interactions. Non-bonded has like Leonard-Jones and Coulombic. So charge groups here are interact through both LJ interactions and Coulombic energy potential. They also use a cutoff, which is generally at 1.2 nanometers. So anything above 1.2 nanometers, they don't consider. So this also increases the computational efficiency. So moving on to the bonded interactions, these are the equations that represent the bonded interaction. So VB here is for bonding between two beads, for example, where KB is the equilibrium constant. So the bonded potential and all the bonded interactions, they are calculated by matching them with PDB distributions. So I'll explain them, how the PDB distribution is calculated in the next slides, I think. So this is like for all the amino acids, how many beads we can represent. Since glycine and alanine are smaller, they are represented just by one bead, whereas like bigger beads will have a polar along with polar bead in the end. So they still retain like chemical nature. So in like considering the mapping of amino acids, so if you consider this as one chain and this one amino acids with side chain S1, this is the B means backbone bead. So this is the backbone structure. So based on where the backbone is present, whether it's in helix or beta sheets or elsewhere, coil or bend, So we assign different kind of interactions. So this DBB and the theta values, the angle between and the dihedrals and the side chain angles all vary depending on where the amino acid is present in the secondary structure. If you consider proline, for example, it is less polar due to lack of hydrogen bonding capacity. So if proline is present in helix or beta sheets, the hydrogen bonds reduce the polar character significantly for proline. So this is how the parameterization for bonded interaction are done. So all of the bonded parameters are optimized based on the PDB distributions of bonds, angles, and dihedrals. So I think they consider around 2000 PDB proteins to measure the bond lengths and the bond angles. So if you can see this distribution here, helix is represented in solid, a coil is represented in dashed, and the less gray color, I think that is represented in the extended form. So again, depending on where the secondary structure is, we assign, we calculate the equilibrium constants required for MD stimulations. When I say equilibrium, so these are the values that we need. That's how they just like measure it through PDB distribution. So what are some of the tools for using MARTINI? Is there's some tool called insane.py, which can pack a membrane proteins, which we use. This is like this is for a fast receptor, which is packed in the lipid, and I remove water just for easier viewing. And there is another tool called MARTINIze, which is to coarse grain the protein. So keep in mind that like we don't have to do any of this manually. For most of the standard protein structures, the MARTINIze and insane tools can coarse grain and also write topologies for you. In addition, we can also add elastic network. We use elastic network in coarse-grained simulations because we want to keep secondary structure and stable secondary and tertiary structures of protein in place. So moving on, I want to show some of the results that we produced using MARTINI. None of this is from our work, but I've just put in this figure to explain like the motivation. So we all know about COVID virus, which impacted lots of life last, I don't know, 3 years. So, so the main pathway through which the virus interacts with us is through the spike. This is the spike of COVID-19. It is divided into two domains, S1 and S2. So just the science behind here is, so there are two states when the spike protein interacts with ACE2. ACE2 is the enzyme found in our body. The spike interacts with ACE2 enzyme. So the ACE2 enzyme through furin proteases, it removes the S1 and S2 subunits, and that's how S2 enters into host membrane. So now we divide this form into two states called prefusion state. Prefusion is the S1 sub. So S1 subunit that contains receptor binding domain. Receptor binding domain is where most of the ACE2 molecules interacts with the RBD. RBD is the receptor binding domain of the S1 spike. So what we need for vaccines or antigens is that we need a stabilized prefusion conformation of this fusion protein because it has most of the neutralizing epitopes and that can be targeted by antibodies. In general, the perfusion state, if we know, if we can like stabilize the perfusion state in antigens, our body can better recognize it in its structure. So this is the post-fusion state. We for antigen development, we would want to suppress the post-fusion state. So just like how the vaccines are, why we are doing this current work. So our collaborators, we are trying to produce antigens which are stable in the perfusion form. So that's why I just introduced some concepts. So how can we stabilize the protein in the perfusion form is through different strategies. One is through we can introduce salt bridges, we can introduce cysteine bridges that keeps the structure stable. So recently we use 2 proline substitutions in the S2 subunit as indicated here. Prolines in general stabilize the protein structure. So it's exactly where it is S2 subunit in a turn. So proline in general stabilizes the perfusion form of the protein. So if we consider the entire virus, it has like 305 million atoms, including 24 spike proteins. If we just consider the lipid bilayer along with the spike, it has like 1.7 million atoms. And the spike with ACE2 in the glycosylated form has like 8.5 million atoms. That is extremely hard to simulate with for like our interactions where we need like microseconds of simulations data. Another thing I wanted to introduce is it's heavily glycosylated in terms of different glycoforms. So in this figure here, N17 is the residue position where we can see these many glycans. Some of them will be abundant, but similarly, there are like various other glycoforms that are found. So computationally adding all this and simulating them individually, so there's like huge computational time we need to calculate all this. So our approach was to use coarse graining, that is through MARTINI simulations. And we right now are only trying to add glycans, which are highly abundant. Which one among them have like highest abundant? So with that, our modeling approach is first we take the PDB structure that we get. We already have the PDB structure for 6VSB is the one. And we model the missing residues and loops through modeler. If you want to learn modeling, this is one of the important tools that can do a lot of structure modifications in terms of like if you want to substitute amino acids or to build loops, to build extra transmembrane domains and things like that, you can use modeler. And to attach glycans, you can also use CharmGUI, or there's one more website called Glycam. So you can produce the results which minimizes the energy, but since that is a server-based website, sometimes it fails. And in our case, we have like 22 glycosylation sites in one of the protomer. So 22 into 3 is like 66 plus some glycans on the ACE2. So it always fails. So in our group, we actually have inbuilt script, like in-house script that can energy minimize glycans called glyprot, written by Austin, I think.
Yes. It's
Yeah.
actually published by now.
Yeah, yeah, it's published. It's available if you want to energy minimize the huge structures and glycans, and it works for glycosylated proteins. And next we coarse grain using MARTINI force fields. So for proteins, there is already a script called martinez.py. And for glycans, this is what I worked on. I have automated the process where we can write topologies for all of the glycans. Next is we neutralize, solvate, and equilibrate using bromats. And then structural studies. These two parts I won't be showing today, but I'll be showing pooling simulation results. So this is the protein model. First, we take the 6PSB and we build the missing residues and loops using modular. So some of the residues are different from the sequence that we got from our collaborators. So as I mentioned, modular is the best tool for you to play with the structure, and we energy minimize it.
I have a quick question.
Yeah.
Is there a reason why you use 6PSB?
Because it has like a 2-proline substitution, which is generally stable in prefusion form.
Oh, okay.
Compared
we
to
didn't do the proline substitution ourselves. We're using the PDB that already has proline substitutions.
no. So that has like a minimal difference in terms of sequences 6PSB compared to other form. And we also needed to model ACE2 on the RBD upstate in the chain A form. So 6VSB was mostly to go for us.
Oh,
Yeah, but we did the proline substitutions here.
Oh.
Yeah. So coarse graining the glycosylated model. If you ever want to coarse grain a new molecule, if it's whether glycan or any other molecule, there are different strategies. One is to there's like a lot of data already published on how to coarse grain or the force fields for different fragments of molecules. You can either use that, or you can also generate these values by yourself by doing atomistic simulations and matching the equilibrium constants. So what general strategy I used here is to split the molecule. Yeah. So you run simulations long enough, right? You start with a dumb value, any value, you iterate and you try to produce experimental results. If it matches, you try to match the values like this distribution, for example, until you get a preferred experimental value. You just reduce the error. So run atomistic simulations.
So you're directly calculating the equilibrium constant, or you're calculating some proxy value from which you calculate.
Oh, I didn't calculate any of those for the protein. It's already there. So I am not sure actually if they actually calculate the equilibrium constant.
Don't calculate the equilibrium constant to calculate the equilibrium distribution. The coarse graining normally care about the distribution. For example, if you have a typical case, typically a torsion. Say you have a simulation, you're measuring the torsion distribution atomistically, and you actually then try to optimize the distribution, not the equilibrium constant. What you see here is all distributions. It's all probabilities.
Not the one-to-one per se, but it's the distribution.
Normally, always there are a number of different ways. Often is one way. There are other ways.
Yeah.
But essentially, all coarse graining approaches are normally going structural to optimum to reproduce atomistic or experimental structural distributions. So that your conformation, your structure on the large scale is the same in the atomistic simulation and the coarse-grained.
In the large scale, it's distribution again, it's not one-to-one. Because you also have reduced number of degrees in terms of
Yes,
the
you only can of course look
atoms.
at distributions of coarse-grained, not of atomistic degrees. So what you do in that case, you do a core atomistic, if you don't have this, you do an atomistic simulation, you analyze it as if it was coarse-grained form. You do the mapping, you do this, you take the map, you do the structure of the mapped structure, and then you optimize the coarse-grained potential such that that mapped structure is represented.
Yeah, there's another tool which backpropagates. In the sense you have done coarse graining and done simulations, you can reproduce that back to atomistic structures, like divide the bead into 4 atoms.
That of course is not unique. That is a one-to-many, yes.
Yeah, one too many. Yeah, so in general, most of the, if you have a new molecule which you want to coarse grain, if you want to study like a larger system, larger biomolecular systems, I would suggest look for literature and find there are like fragments of the data. For example, you might just find data for this part and this part, and so you can try to simplify the model, and you can create a topology by modifying the existing file. Similarly, then you can optimize further by your normal process of MD simulations. You minimize the structure, then set up simulation with multiple copies, and then you try to refine the model by adjusting the parameters in terms of you can vary. Okay, 19 to 15 maybe not so well. Try to calculate that distribution and try to model. So this is the glycosylated model for our structure. As you can see, I just put protein in the cartoon structure with, I think it's Gaussian surface model for the glycans, just to signify how heavily our protein is glycosylated. So this is the pulling simulations. When I say pulling simulation is steered MD molecular simulation. So you have 2 atoms that are attached to spring and you pull one away from the other. So what it essentially tells us is how great the interactions is between one molecule with the other. So if there are like lots of hydrogen bonding and other interactions, it requires like a larger force to pull them. If it's not a great interaction, like lower in terms of the force that's required to pull them apart, it'll be slower. So this is for our coarse-grained spike. The entire spike with case 2.
So whatever you're pulling here, the RBD is
The RBD from the spike, entire spike. So let me just go back. So this is my ACE2. So this is the RBD up. So I'm pulling them apart. So
just one of the RBDs, right?
just one of the RBDs, because I'm only modeling one ACE2. But yeah, in like next steps, we will have 2 RBDs up and we'll do both.
To further about, does it make a difference in the direction of the move? Like if you're pulling it, let's say the X direction was Y versus Z, would that make any difference in the energy?
Yeah, pulling is like a reaction coordinate. So this is like a box, and say this is my RBD and ACE2, and this is my spike. So you pull it apart in this direction, say it is Z. What do you mean changes?
Let's say instead of pulling it in the Z direction, pull it in the Y direction.
Oh, actually you can pull it like any direction you want, even XY. So here I just pulled it because Z is like the most interactions are along this direction. But yeah, you can pull it like this too.
Technically, what you should do, but you have kind of know which way it goes. Technically, what you should do different directions, and then the lowest one would be the middle one. The normal, you know, are already beforehand what it is. Yes.
And we have like enough literature on like the which is the direction, like these like most interaction.
But if you don't know, you would do different directions and you would go with the minimal. And also it depends on how fast you're pulling. That's not a thing we have to work on.
Yeah.
Because pulling is non-equilibrium. So non-equilibrium, that means it's, as long as it's non-equilibrium, it is
So one good thing that I could produce was like so on the right here we have all atom MD simulations for just the RBD and ACE2 for both glycosylated and non-glycosylated, which was from our group Ihan and Brad worked on this. So we were able to reproduce similar results. Our results indicate that so the glycosylated model leads to stronger and longer range binding interaction between the ACE2 and our spike, which essentially was our group's paper on the all atom MD simulations. Yeah, so right now I'm working still on like optimizing this, like produce enough replicas to see if this really holds good for all the replicas. And then I'll calculate the distance distributions, like in terms of contact maps, which of the key residues that are interacting, especially for our antigen. And I'll be working on another model for the protein called HexaPro, which has like 6 proline substitutions instead of 2p, and which is even more stable. Yeah.
This is a very good information. If you see around 1,000 seconds,
Mhm.
400, let's say 200 or 100.
Mhm.
So is this expected?
This?
Yeah. So
Um,
like the delta is very high, even for the like time which is at 12,000 seconds.
Yeah.
At that point also the delta is very high between different axes.
Yeah, it's the statistical average is what we mostly worry about. The average, but yeah, because MD simulations, we mostly want to calculate we don't worry about individual points here. We worry about statistics like the average. So that
this is a single trace. So we need
more replicas.
multiple simulations.
So what I do is like I take multiple replicas of the system. So there'll be different initial conditions to begin with. So I try to map all of them and like get the average behavior with that.
What you see on the right hand side, there's multiple, for example, multiple blue lines, multiple yellow lines.
Yeah.
They are multiple replicas.
Yeah.
I'm just seeing because the delta there on the right hand are very low compared to the features on the left hand. That's why
The patients are very high.
The fluctuations on the coarse-grained level are higher in this case because the coarse-grained time steps off.
So, okay, I think I didn't mention about the time steps. So for normal MD, it's like 1 to 2 femtoseconds. For coarse graining, we use like 40. So that's why we get 2 to 3 hours of better efficiency.
So it's effectively, you need also more simulations in order, otherwise it is just noise.
Yeah. Yeah, and like I could produce all of them in like 1 day versus I don't know how long this would take.
That was a few months.
Okay. Yeah. Like not 1 day per se, but you know, around that time frame, like within less than like a week.
So yeah.
Yeah, but essentially,
Small system, right?
yeah, it's like much smaller. This is just like just this part and this much RBD. So
That's also
reduce
the
that
part
with
of
that.
the noise because it's much larger system.
Yeah.
Okay. I have to run.
Okay.
Yeah.
Yeah, sure. Yeah, so yeah, the only thing is it gives us semi-quantitative representation. It's not accurate. You just always have to validate with atomistic simulations and known experimental data. to say that, yeah, you're seeing something that,
Capture the mix and match. We have
Yeah.
part of the system being a part of the system being used.
yes, yeah, people have done that, like, especially for membrane proteins. They've done it, like,
So
yeah.
part of the system where it's been a system will be the non-interacting one, right? Other than non-reactive, but I'm not sure how.
No, everything is interacting, but instead of, say, like, carbon is interacting with another carbon, or carbon with hydrogen, you are combining 3 carbons and making this entire bead interact with the adjacent structure called a bead. So you are essentially ignoring this.
So when you're doing the mix and match, like you're having a hybrid system,
Hmm?
what is the time scale? Because you mentioned the most meaning you can use of 14 at the same time scale.
Yeah.
Which way you want to use at the same time scale.
Yeah, even if you use a smaller time step for this coarse graining, your number of degrees of freedom still reduce, because instead of having, say, if your system has like 10 per 6 items, 10 per 6 by 4 essentially reduces the number of degrees of freedom for coarse graining. So your scaling still reduces.
If the order is
No, I think the order is n log n is the dependency, if I'm not sure. For scaling, yeah, for scaling is n log n, so it does reduce.
I think what I understood, fact is that you gain lower time steps, like 0.5 or 1.5 to account for the vibrations between the bonds. Since your core graining that you're no longer we have a higher time step.
No, I think his question was, for hybrid system, what time step would you generally use? I never used a hybrid, but from what I remember, I think they generally use lower time steps because you have a domestic. It still is better in terms of calculation interactions between two, because instead of,
just thinking that that's a more of a
yeah, I'm not sure the mathematical thing behind like how to calculate. I think it's n log n dependency is what I remember with wallet scale.
You're dividing by 4. The order will still remain 10 to the power 5 into the power 6. So that's why I'm saying it won't make a big difference. Like unless until you divide by 100 or like 1000,
Right,
that's when the scales like or reduced drastically.
exactly. So yeah, I'm not sure if it's by 4.
No, I don't think that's.
It's not.
Probably divided by
Yeah.
a larger number.
That's more.
Yeah.
So
Using all those, like larger chunks.
yeah. So in my system, if I want to like show, like I have like a very huge system. I can just like, if you have time, I can show you our sorry.
Just thinking we can do the same thing.
Oh, I don't think that would work because you're doing quantum mechanical and
QMMM is partially there.
Yeah.
You have you basically separate your system into a fluid part and a liquid part, the molecular mechanics on one part, the quantum mechanics on another part. So these are enzymes are different.
and you generally I don't know. Yeah. So this is how big my atoms are. Yeah, I have to share my entire screen, but yeah. Share. Okay. It should count. But can I drag it? Oh yeah, great. So this is like how big. Along with it, I have like I don't know if I can download the box file. So my box is this big. because I have to pull it like this around 5 nanometers. So that's like, I don't know how many atoms. I should have included that, but yeah.
The system is closed.
Yeah.
So what, which kind of design the beads? You mentioned that 3 or 4 atoms are clubbed with one bead, right?
Yeah, based on like the chemical nature. So for example, so we divide based on, for amino acids, there are like polar beads. So if you, I don't know if you know, there are like charged, oh, you're not familiar. Yeah. So amino acids has like charged beads. They have like charges, there are like polar, they have like hydrogen bonding capabilities, which are all like different from one amino acid to amino acid. Also, depending on where the amino acid is present in the protein structure, those values change. So that's how they calculate that based on this interaction matrix, essentially. So for charged beads, for example, they assign like a DA, a bead number called DA in a topology file. If that is present, that has like both. You can,
I'm saying how would you guys happen with have bead X? What do you decide your atoms are going with that?
oh, okay, like how do you know like
Yeah.
which of them?
Exactly.
Yeah, again, they're reproduced based on the experimental part, which of them produce better partitioning free energies. Because yeah, you can combine these 4 instead of these 4. That's what.
How big can be like one, how big, in terms of number of atoms?
Generally, they mostly use only four atoms. Other than that, 2 atoms also for like ring structures, which are like difficult to retain in terms of their shape. Yeah.
I had one question regarding the box. Second number, let's say, showed here, water and oxygen. Yeah, so my question is that is this how the box is exactly?
No, this is after certain time scale, like microseconds. They run a very long.
No, I'm talking about the initial unit set. But I was asking why isn't there a sandwich between the two different phases? Because you can actually have, if you have a system like unit cell like this, then you can actually have outside the box interactions, which cannot be accounted for properly.
You mean like periodic boundary conditions?
Yes, very good.
Oh, I think that I'm not sure how this figure was obtained. This is from this paper workshop. But I think they do apply periodic boundary conditions. Sometimes you just don't correct for the periodic boundary conditions.
That was my question. Like, because you're having periodic boundary conditions, you're not technically accounting for a second interface, which is happening here.
Where? Here.
So here, if you look at just the unit cell, you have a single interface between water and oxygen.
Right.
But
Oh, this interface.
yes, the interface between the phases, because it's a biphasic system.
Right.
So, but technically, when you consider only a particular unit cell, you are considering only one interface, whereas actually you are having 2 interfaces according to the boundary conditions, periodic boundary conditions. So why isn't there why did they not use a sandwich model where you have basically one phase, like phase 2, phase 1, phase 2 in a single unit cell, so that you know that you are calculating for 2 different.
Oh, from exactly here.
Yeah.
So firstly, I don't think I know if they actually use the PBC here. Okay.
But,
Secondly, yeah, I am not technically sure how they, if there is any thing based on the PBC conditions, like
because if the outside box interactions are very, very large, then you can actually see some globules of water in octanon phase or some globules of water on the bottom.
You mean interaction from this PBC box to this one?
Yeah.
Yeah, that could happen.
The unit cell will be a very something which can be really invisible.
That could happen too, but again, I'm not sure what conditions they used here. I have to look into that paper actually. Yeah, that's a good question. I'll keep that in mind. Might come up in my QE.
Um, Then, yeah, let's thank her for her presentation.
Thank you.